So the discovery that DNA carries genetic information is actually sort of an interesting history lesson. And it's fascinating actually to look at the experiments that led up to the discovery that DNA is actually what we inherit cell to cell and generation to generation. And it's actually placed in a very interesting time in history as well. And so we want to take a look at it. Really what started the kickoff leading towards looking at the macromolecule DNA as the genetic information actually came out of the influenza pandemic in 1918. So governments all around the world were really racing to try to find a vaccine for this influenza strain. And there was a microbiologist named Frederick Griffith in England, and he actually found that a substance in a heat-treated virulent strain of Streptococcus pneumonia, which was causing the flu, could transform a non-virulent strain. So he was looking at and trying to investigate how to treat the Streptococcus to make it non-virulent. And he actually picked up and observed that non-virulent bacteria could actually be transformed and become the virulent strain. And he wanted to know how that was happening. And so he did this very simple and elegant experiment. So he took virulent Streptococcus strain, injected it into mice, and the mice died. When they dissected the mice, they found the living strain of the bacterium in the heart of the mice. However, if they did that same experiment with a non-virulent strain of the bacterium, the mice live and they're happy. There's no bacteria in their hearts. So when Griffith heat treated the S strain and then injected it into the mice lived. And so he thought that that was interesting. His next experiment, and I don't know how he decided to go from just heat killing the bacteria to mixing the heat-killed virulent bacteria with the non-virulent strain, but he did. And so when he mixed the two together then, the dead S strain with living non-virulent strain, what he found was that the mice still died and he could detect living S strain back in the hearts of the mice. And so he concluded then that there's a chemical substance from the dead cells that was transforming the non-virulent bacteria into virulent bacteria. And some of the things that he didn't know at this time that we now know and we can see influencing this experiment is that first when DNA is heated up and then if it cools, it can actually renature. And the other thing that we now know is that bacteria can take up nucleotides from their surroundings. So that's what we actually still today call transformation. Bacteria taking up nucleic acids from their surroundings. So a group led by Oswald Avery thought that this was interesting information. And in 1944 then, he did an experiment to look at, well, what in this virulent strain is it that is transforming the cells? He was studying macromolecules, and so he was learning a lot about the different macromolecules that are on actually the outsides of cells. And so what he did was he heat treated the S strain, so the same Streptococcus bacteria. And then what he did was he took that liquid and then he treated it with a bunch of enzymes. So he took all that bacteria, he treated it with enzymes. And the enzymes he put in were protease, and so he destroyed all of the protein that were outside of the bacterium. He put in RNase, he destroyed all of the RNA, and DNase, so he destroyed all of the DNA. And then he took all of that treated S strain bacteria, He mixed it with a non-virulent strain, just like Griffith did, and then he looked to see if transformation occurred. So could the R strain pick up on and become transformed? And what he found was that without protein around and without RNA, the transformation could occur. So new S cells, the virulent cells, appeared from the non-virulent cells. However, when DNA was absent, that transformation did not occur. And so in that culture, all he detected were R cells. There were no S cells basically brought back from the transformation. And so he concluded that, well, it was indeed the DNA molecule that was responsible for that transformation. And just like many things in science, at the time, everybody in 1944 thought that proteins actually had to be the genetic carrier of information because proteins were so much more complex than DNA. And so the scientific field didn't accept his finding. They did not think that DNA could actually be the genetic information. And so it wasn't until quite a few years later then that two more scientists named Hershey and Chase, they did an experiment to prove that it was not protein. That was the hereditary material. And so their experiment was also very, very elegant. They were working with a virus called a bacteriophage. So bacteriophages actually just infect bacterium. And so they knew that a bacteriophage could infect a bacterial cell, and then once the cells were infected, the cells actually lysed and they released more phages. And so they knew that the phages were passing on hereditary information to produce more and more phages that grew inside the bacteria. And so what they did was they wanted to see, well, is it phage protein or phage DNA that was the hereditary information? And so using what they knew about protein and DNA, they actually labeled the two differently. So proteins have amino acids that contain sulfur. So they put radioactive sulfur and mixed it with the phages so that the phages incorporated the radioactive sulfur into their proteins. And there are no phosphorus atoms in DNA, so it only labeled the proteins as radioactive. And what they did then was they allowed these radioactive phages with their labeled proteins to mix with the bacterial cells to infect them. And then they blended bacterial cells so that they could knock off all of the phages that would be attached to the cells. And then they centrifuged the cells to pellet the bacterium, and they got rid of all of the supernatant that would have had the phage excess proteins. And so they looked then just at that bacterial pellet, and they did not detect any phage protein with the bacterium. Then they did the same exact experiment, but instead of labeling proteins, they labeled the DNA, and they used radioactive phosphorus so that would incorporate into the sugar phosphate backbone on DNA. So here again are the radioactive phages, but now the DNA is radioactive. They allowed it to infect the bacteria cells. They agitated by blending those cells to knock off all of the phages. They pelleted the solution then, so at the bottom would be the bacteria, and they got rid of all of the excess phages. And at the end, they did detect radioactivity in the bacterium that were left. And so that meant that the phages, when they infected the bacterium, they were injecting their DNA. And so the DNA was what was the hereditary material. And so then building on these experiments, another group came up with the theory then that genes were directing the protein synthesis, and they came up with what we call the one gene, one enzyme theory. And so two scientists called Beadle and Tantum, they made a connection that if a gene is mutated, then we have non-functional enzymes. that result. And so they started to link that there, for each gene, there was a non-functional enzymes. And so what they did was they actually took bread mold spores and they irradiated them with X-rays. And those X-rays then caused mutations in the DNA. And then what they did was they tried to grow the spores on minimal mediums that were missing molecules for making arginine. And they isolated 4 different colonies that had a mutated gene in them for part of this biosynthesis pathway. And what they found was that they could add back in precursor molecules if it was a step after the mutation. And so from that, they came up with this concept then that if there is a mutated gene, then an enzyme doesn't work. Today, what we usually say more accurately is that each gene actually specifies the production of one single polypeptide or a protein component. What we now know with protein structures is we can have quaternary structures where multiple polypeptides come together to form that protein. So then in 1958, Crick came along and he wrote The Central Dogma of Molecular Biology, and we still stick to this today. And so this is what most people are familiar with, and we introduce it in all beginning biology classes. Now we know that DNA is the hereditary information that is passed on through generations of cells, and that from this DNA we make an RNA template that's brought out into the cytoplasm of the cell. Transfer RNAs match from the messenger RNA codon to their anticodon, and these transfer RNAs carry with them then and match up amino acids. And as they do so, we start to make a polypeptide chain through the process of translation.